Hello, my name is Kimberly Michelle Madero Peñafia. We want to talk about determining the specific role of extracellular exosomws in the interaction between epithelium and mesenchyme during tooth developing. Definition: what exosomes and why are they important? Exosomes can be described as extracellular vesicles. The term exosomes was first described 30 years ago as small bubbles, with a diameter of approximately 14 or 100 am that can be secrete most cells. These cells have a lipid membrane structure and internally coated with proteins, mRNA, microRNA, and the other substance. In addition to exosomes, there are also the other types of microbubules in cells such as apoptic body and extranuclear particles. Each type has different functions. Exosomes are often overlooked as separate category, but they are functionally different types oversight. Mesenchyme. Emerald is derived from the ectoderm of the oral cavity. The remaining tissue different from subrone mesenchyme derived from the mesoderm of the neural crest cells. Cells in the neural crest receive morphogenetic information before or shortly after migrating from the neural crest, as the upper and lower jaw grows and the accommodate developing teeth. The shape of the face change odontogenesis to developing, begin with the inducing influence of the neural crest mesenchyme on the overlapping ectoderm. Epithelium-mesenchyme interaction. This interaction, especially the interaction between epithelium and embryonic mesenchyme, play as a crucial role in the intuition, the development, and the differentiation of most, if not all, of the organs of vertebrate embryos. These interactions are classified into three categories. 1. Those in which the epithelium controls the differentiation of the mesenchyme, development of the kidneys, sometimes a neural crest, and 2. Those in which the mesenchyme controls the differentiation of the epithelium, development is the hair and the salivary glands, and 3. Those in which there is reciprocal interaction between the epithelium and mesenchyme trigger outline in the limbs.
The session was centered on the biological properties of mesenchymal stem stromal cells. This is a particular type of cells which has unique properties because it has the ability to generate an immunomodulatory environment that is very beneficial in situations in which there is a rampant immune response, like in the case of graft-versus-host disease, but also several other autoimmune diseases. So what we found in particular is that the mechanism of action of these cells is largely based on the ability of these cells to undergo cell death when they are injected in vivo. So what we found is that the recipient of the cells is actively killing these cells, and as a result of that generate a benevolent immunomodulatory environment which is very beneficial in these type of diseases. Based on that, we can now even predict who are the patients who are going to respond, because only those who kill the cells are the ones who are going to respond to the treatment.
Application in odontology. The dental pulp system cell or the English dental pulp system cells (DPSC) are found in the central chambers of the dental cavity as the soft tissue that infiltrated by a network of blood reveals in the nerves that emanate from the apical formation of the tooth.
Our bodies contain many specialized cells that carry out specific functions. These specialized cells are called differentiated cells. In contrast, a stem cell is undifferentiated. Its role in the body is not yet determined. A stem cell can give rise to multiple cell types, including more stem cells. Stem cells play an important role in our bodies, from embryonic development through adulthood. Development begins when sperm and egg meet. This event produces a special kind of stem cell that has the potential to grow into a human being, as well as a placenta, which will nourish the embryo as it grows. For the first few divisions, all the cells remain undifferentiated. Each one has the same potential as the fertilized egg. As development continues, a series of instructions, or signals, gradually limit each cell's potential. Differentiation has begun. 1 week after fertilization, the embryo, now called the blastocyst, looks like a lopsided hollow ball. The cells around the outside of the ball will form the placenta. A clump of cells on the inside, called the inner cell mass, will form all the cells of the body. About 2 weeks after fertilization, the cells organize into 3 layers. Cell signals restrict the cell's potential even further. Each layer will give rise to a different set of cell types. A few weeks later, the cells of the outer layer, or ectoderm, have become skin, nervous system, and face and neck. The middle layer, or mesoderm, has become muscle, blood vessels, and the beginnings of bones and connective tissue. The inner layer, or endoderm, has begun to form the digestive and respiratory tracts and the glands that feed into them, including the pancreas and the liver. As development continues, cells differentiate to form the 250 or so cell types of the body. But even after birth and into adulthood, pockets of stem cells remain. These somatic or adult stem cells play an important role in growth, maintenance, and repair. Some stem cells sit quietly, waiting for a signal to tell them to divide. Others are constantly at work, replacing cells that are lost through daily wear and tear. Somatic stem cells differ from embryonic stem cells. Under natural conditions, they can give rise to just a few cell types. For example, bone marrow stem cells give rise most often to blood cells. Bone marrow contains massive numbers of stem cells, but most somatic stem cells sit among large numbers of differentiated cells. Somatic stem cells have been found in many other tissues, including brain, blood vessels, muscle, skin, teeth, heart, gut, and liver. As we grow from fertilized eggs into fully formed human beings, stem cells give rise to all of our differentiated tissues and organs. Stem cells continue to play an important role throughout our lives as they
Pros and cons. Pros: the mesenchymal cell could be considered a cell capable of the proliferation and differentiation into mesodermal and non-mesodermal cell lines, including clonogenic, osteogenic, myogenic, and adipogenic. They have the potential is a therapeutic alternative for the managing disease characteristic by acute or the Crohn's disease loss. Cons: when there is an inadequate genetic expression, alternative in organogenesis occur and, depending on the affected genes, absence or malformations can occur, causing defects such as regional odontodysplasia, which some authors associated with a more points mutation, but the other authors' etiology has been attributed to local trauma or systemic factor, being low prevalence pathology, and the origin is not yet well established. The regional odontodysplasia (OR) is a non-hereditary developmental disorder that affects both the primary and permanent teeth and involve the mesodermal and ectodermal tissue of the tree, with the condescending with clinical radiographic histological findings. Issue applied in Ecuador. Clinical application in Ecuador is having these cells. The process for the installation of the oral mesenchymal stem cell storage form and cryogenic is possibility application of the field of regenerative therapy remained for the analysis its potential and regenerate the periodontal ligament has also been demonstrated is a promising treatment for the periodontal disease and its consequence, and the possibility of beginning use in neural regeneration, where there are divided opinion on that part of scientific community between those studies that mention the possibility of generate functional neurons from adults MNC, and those who recognize that this property is not such is the type of cells. They possibly use the stem cells through this way enabling in functional replacement for patients suffering from xerostomia. I also postulate.
So a mesenchymal stem cell is a cell that can become cartilage, fat, bone, blood vessels. Other people are also trying to drive them, for example, into insulin-producing cells. We have investigators in this institute doing that. So they have a lot of potential. But the most intriguing part to me is their ability to limit inflammation. When we transplant islets, in the pancreas, they get a great deal of the blood flow. They have a lot of vascularity. And when we transplant them, they don't have functioning blood vessels. And we know that in our preclinical models, it takes about a month for the blood vessels to develop. So the concept was knowing that these mesenchymal stem cells can actually stimulate vascularization, that if we transplanted the islets with the MSC, we get vascularization more quickly, thereby allowing the islets to function faster. In addition, we know the cells are anti-inflammatory. They limit early inflammation that occurs when we first put islets in the body. That inflammation, anytime something invades the body, a splinter, you get redness, you get heat, it's inflammation. And that's a non-specific effect of the immune system. And we know that MSC can limit that. So the idea that we could increase vascularization, limit the inflammation that we know occurs early post-transplant was very attractive. And the hypothesis is that when you give them in a blood vessel, that they migrate to the site of inflammation. And so we believe that they migrate to the liver where the islets are rejecting, limit the inflammation, and then stimulate migration of stem cells into the air.
In conclusion, the possibility that stem cells offer both in the field of the dentistry and regenerative medicine are varied but still very little studied or clinical tested. This is hard-held advance is field of historic science is presented in the next step in medicine and the therapy of disease that today is difficult to access quick recovery. This is necessary to know more must be to define the protocols for clinical application this new opportunity that is the percentage used, for example, studying how the cells also transplant in the body, decide their fat between being part of the solution, self-renewal, and differentiation of part of a new problem, carcinogenesis, and developing the mechanism to control this component.
Hello, my name is Li Xiao. I'm a lecturer of the Nippon Dental University in Japan. Here I'm going to introduce my article from regenerative dentistry to regenerative medicine, progress, challenges, and potential applications of oral stem cells. I hope you enjoy it. From regenerative dentistry to regenerative medicine, progress, challenges, and potential applications of oral stem cells. Oral stem cells include oral epithelial stem cells and oral mesenchymal stem cells. These cells can be isolated from oral tissue, including teeth and their surrounding or supporting tissue. In the daily dental clinical practice, there are many oral tissues exist from patients. They are usually disposed of as hospital waste. In Japan alone, about 11 million teeth are disposed each year. However, these teeth and their surrounding tissue are not useless. They are treasures because we can get stem cells from them. Especially the deciduous teeth are more valuable because they can provide higher quality stem cells. Here is the human third molar, wisdom tooth. When you cut the tooth like this, you can get dental pulp like this. From the dental pulp, we can easily obtain oral mesenchymal stem cells. The tissue attached to the surface of this tooth also provides oral stem cells. These oral stem cells are not only applied in regenerative dentistry, but also widely used in regenerative medicine. Here, I will talk about the current research about oral stem cells. Before we talk about oral stem cells, let's take a look about stem cells and organ formation. In an organism, all the tissues are built from pluripotent embryonic stem cells. The embryonic stem cells differentiate into multipotent stem cells, including epithelial, mesenchymal, and other tissue-specific stem cells. Interactions among these cells initiate and regulate developmental processes, resulting in the formation of highly specialized functional tissues and organs, such as tooth, hair follicle, lungs, kidneys, and other tubular organs. Once the organism matures, the pluripotent embryonic stem cells are vanished, and some multipotent adult stem cells remain in the developed tissue to sustain the homeostasis and repair injuries. Adult mesenchymal and epithelial stem cells remain in oral tissue. Here is a diagram of an immature tooth. From dental pulp, gingiva, and periodontal ligament, we can obtain mesenchymal stem cells, including dental pulp stem cells, DPSCs, stem cells from exfoliated deciduous teeth, SHED, stem cells from apical papilla, SC.
Thank you.